It is my pleasure to introduce today's speaker, Dr. Vivek Sebaya. Dr. Sebaya is an associate professor in the Department of Investigational Cancer Therapeutics, associate professor in Division of Pediatrics, the clinical medical director of the Clinical Center for Targeted Therapy, a phase 1 clinical trials program, and executive director of cancer medicine research at the MD Anderson Cancer Network. He served as the principal investigator in over 50 phase 1 or 2 trials. His research interests include drug development in rare cancers, including first-in-human and practice-changing novel basket trials. He co-led the first histology-independent international vemurafenib basket trial in non-melanoma BRAF V600 mutated cancers. This led to US FDA approval for use in Erdheim-Chester disease. He served as the PI for the Global Rare Oncology Agnostic Research, or ROR trial. Dadrafinib and trametinib was US FDA approved in the treatment for BRAF V600 mutant anaplastic thyroid cancer based on data from the ROR trial in 2018. He also led the 2 selective RET inhibitor trials, selpercatinib and pralsetinib, for RET-dependent cancers. These trials both received FDA approval for RET-positive lung and thyroid cancer in adult and pediatric patients. He co-led the lurbinectedin basket trial, FDA approved for small cell lung cancer. In addition, he led a phase 1 trial for pemigatinib, an FGFR inhibitor for FGFR-positive cholangiocarcinoma. Furthermore, his efforts laid the groundwork for therapeutic breakthroughs in the field of sarcomas, including for a trial for radium-223 as a treatment for relapsed osteosarcoma. And this trial was singularly responsible for the inclusion of radiopharmaceutical in the treatment algorithm for osteosarcoma in the NCCN guidelines. He's a major advocate for precision oncology. His national leadership role continues to expand as the national PI for BRAF non-V600 alteration arm of the NCI Match Precision Medicine clinical trial, and is a member of the NCI CTEP PTMA for a glutaminase inhibitor basket trial. He's published over 200 peer-reviewed articles in several prestigious journals, including the New England Journal of Medicine, Journal of Clinical Oncology, and Nature Reviews Clinical Oncology, to name a few. Please join me in giving a warm welcome to our prestigious speaker, Dr. Subbiah.
Thank you, Michael, and everyone for being here. It'd be a pleasure and honor to see you all virtually. Hope to see you all in person post-pandemic, and we keep our fingers crossed that there is not another surge with us. All right, so moving on right into the topic, from tissue-agnostic to NF1 therapies, revolution of the precision oncology paradigm. For full disclosures, I will discuss off-label and investigational use in my presentation. So in the brief agenda for today, I'll be talking about precision oncology and personalized medicine. Again, what is it and why we need new drugs in oncology development, and also share with you my perspective on some trials, clinical trials like the BRAF and RET that recently received FDA approvals, and my perspective and my brief experience with doing these clinical trials. So why do we need new drugs, new approaches, and new tests for treating cancer? Again, before COVID-19, cancer was public enemy number 1. And it will be the public enemy number 1 after COVID-19. And we need to think about cancer like COVID-19. I think that now the world, the general public also has an understanding of what mutations are, what, how a disease can, impact humankind, like COVID-19. So cancer is something similar to COVID-19, although they don't see that in as a communicable disease in the impact. So if we keep the momentum, we will have licked the cancer problem in 1,200 years, even with the same momentum that we have precision therapies and immunotherapy. What can patients expect from the widely FDA-approved drugs that are used? Again, these are the highly sought after, build treatments across the board. Like, for instance, gemcitabine, bevacizumab, timodar, docetaxel, these are the cetuximab, these are the most commonly used drugs. And if you see the survival gain from these drugs, it's in the order of weeks to months. And the response rates to these widely approved drugs vary from 2 to 20%. So we can all do better. I think each and every one of us, it's up to us to think about how we can improve and move the cancer needle. So how did we treat advanced and metastatic rare cancers before the era of precision oncology, before the era of biomarker targeted therapies? Today's reality is metastatic epithelial malignancies remain incurable. Statistical victories with p-value less than 0.05 are hailed as major breakthroughs and advances despite survival gains of weeks to months. Enter precision medicine. So what is personalized medicine or precision medicine? The management of a patient's disease or disease predisposition and administration of therapies that convey maximum effectiveness and safety given the patient's unique genetic and molecular profile. From the current practice of one treatment for the average patient to customizing the right treatment for the right person at the right time, we are still yet to realize the promise of precision medicine. Then we have immunotherapy. Of course, we know that the immunotherapy recognizes the mutanome. Molecular anomalies such as PD-L1 amplification are seen in Hodgkin's disease. And there are mismatch repair gene defects in colorectal and other cancers. And of course, we recently have a high tumor mutational burden emerge as a biomarker for immunotherapy. We have a host of immunotherapy targets beyond checkpoint therapies, beyond PD-1 therapies, like ICOS, like LAG3, TIM3, OX40. And we have a lot of kinases, and we have a lot of kinase inhibitors. All the monoclonal antibodies, all the small molecule inhibitors. So our role, to bring new drugs to patients with cancer, navigating the roadway, navigating the funding space, staff training, overcoming the challenges of academia, industry, working with NCI, working with new regulatory issues, working in the context of challenging insurance environment, working with an evolving landscape and offering multidisciplinary care. So these are some of the targets for therapies. And the next set of slides will be talking about basket studies in oncology, also touching down upon the Memorabilia basket study and the rare cancers oncology agnostic research, and talking about RET inhibitors in clinical oncology. So what are novel precision medicine designs? Again, for the fellows out there. So basket trial, what's a basket study? You need to think about basket study and umbrella study. Basket trial enrolls patients with the same genetic mutation, whether their cancer originated in the lung, breast, colon, liver, prostate, or any other organ. And then has the patients receive all the same novel treatment that targets a specific biomarker. This strategy highlights a different way of thinking about cancers, not based on the tissue of origin, but rather the mutation that drives them. An umbrella study is something similar, but it's a histology-dependent mechanism. For instance, umbrella trial would one type of cancer, for instance, lung cancer, with different genetic alterations. The trial would enroll different patients with different mutations. So umbrella trial is one type of cancer, different genetic mutations. Basket study is one common genetic mutation, multiple cancers. Again, you get this idea. So basket study, we screen patients and we enroll patients based on mutation A, B, and C. So the next set of slides, I'll be talking about BRAF in cancer. Why BRAF? BRAF mutation oncogene is seen in 5 to 10% of all human malignancies. It leads to constitutive activation of the MAP kinase pathway. The most common mutation of the BRAF is the valent glutamic acid substitution at codon 16. It's a driver mutation in multiple solid tumors beyond melanoma, like colorectal cancer, papillary thyroid cancer, non-small cell lung cancer, ovarian cancer, among others. And in heme malignancies, like Langerhans cell histiocytosis, or 100% of hairy cell leukemia patients. So because BRAF was seen across multiple tumor types, we designed one of the first basket studies called the Memorabilia basket study. And again, the theme and the concept was the first basket study that the patients, irrespective of their tumor type, who harbored a BRAF V600 alteration, would be enrolled in different baskets. Initially, we saw that in the non-small cell lung cancer cohort with BRAF mutation, the response rate was 42%. And in fact, we saw head and chest disease or lung and histiocytosis harbored BRAF, the response rate was 43%. This subsequently led to the FDA approval based on this particular clinical trial. Beyond these cancers, we saw responses in rare cancers like anaplastic thyroid cancer, pleomorphic xanthoastrocytoma, cholangiocarcinoma, salivary ductal cancer, ovarian cancer, and clear cell sarcoma. At the same time, we saw 0 responses in colorectal cancer. This was because there was a Nature paper at the same time that showed that EGFR is an innate mechanism of resistance to BRAF inhibition in colorectal cancer. So we amended the protocol to include cetuximab for the colorectal cancer, and we saw responses in that cohort. This led to tumor-agnostic sensitivity of vimorafenib colorectal cancer. So as you can see, we saw responses in multiple tumor types. We saw responses in several tumor types, and this led to the FDA approval in such a rare disease. We recently showcased the pan-cancer efficacy of monotherapy BRAF inhibition in BRAF mutant non-melanoma cancers. So as we know, in melanoma, the science moved on at the same time, and we knew that in the MEK inhibition, MAP kinase pathway resistance was an innate mechanism of resistance in BRAF. Although we saw phenomenal responses with monotherapy BRAF, like agents like vemurafenib, dabrafenib, and encorafenib, immediately within 4 to 6 months, patients started acquiring resistance mechanism through MAP kinase pathway. So the rational target was combination with the MEK inhibition. So this delayed and prevented the potential development of resistance, and patients could stay on for longer period of time with the combination therapy. In addition, the combination therapy also delayed the hyperproliferative regions. With monotherapy BRAF inhibition, we saw cutaneous squamous cell cancer that arose from a paradoxical activation of the RAF, CRAF, and RAF pathways. So this MEK inhibition or MAP kinase pathway inhibition attenuated that process as well. So based on this, we designed the second generation iteration of the basket study called the ROAR basket study, or the rare oncology agnostic research. Again, the concept was similar. Any patient with a rare cancer listed here, like anaplastic thyroid cancer, biliary tract cancer, GIST, germ cell tumor, low-grade glioma, high-grade glioma, including glioblastoma, as long as they harbored a BRAF V600E alteration, they could receive dabrafenib and trametinib. The primary endpoint was objective response rate. So initially, as you all know, anaplastic thyroid cancer is one of the most lethal cancers known to us. The median survival is close to 6 months. And we saw that a subset of anaplastic thyroid cancer patients harbored the BRAF V600 alteration. And we saw the more patients we tested for this mutation, the more patients we found. An initial data set, an interim analysis, showed a response rate of 69% in these patients. And there is no FDA-approved drug for this cancer. And it was, and given the early promising activity in these patients started doing well. Based on a data set of less than 25 patients, this combination received FDA approval. This was the first FDA-approved treatment for this aggressive form of thyroid cancer. So again, with successes in precision oncology basket studies from less than 25 patients, one in head and chest disease and another in anaplastic thyroid cancer. Again, this shows the power of precision oncology, right? Even we can make a difference in these rare, extremely rare orphan subpopulations of diseases. Most recently, we also showed the objective response rate in biliary tract cancer who harbored BRAF V600E alteration. Again, biliary tract cancer patients, 2 to 3% of patients harbor the BRAF V600 alteration, and these patients do respond to combined BRAF and MEK inhibition. So routine testing for BRAF V600E mutation should be considered for all patients with biliary tract cancer. The next set of slides, I'll be talking down upon our recently approved hot RET inhibitors. We'll talk about RET aberrations in oncology, also talk about RET alterations in non-small cell lung cancer, RET alterations in medullary thyroid cancer, talk about why we needed selective RET inhibitors, because we did have multi-kinase RET inhibitors before that, and also talk down upon recent updates. So RET is a receptor tyrosine kinase required for normal development. Alterations in RET function and signaling can lead to tumorigenesis. RET is activated by two major mechanisms. One is oncogenic RET fusions, and one is the activating mutations. Oncogenic RET alterations have been identified in numerous cancers. RET is one of the first oncogenic kinase fusions cloned from an epithelial tumor, and since has been found to be an oncogenic driver primarily in solid tumors. In 2012, right, note this timeline, RET fusions were identified as oncogene drivers in non-small cell lung cancer. And 2017, we had in the clinic entering of not just one, but two selective RET inhibitors. And 2020, last year, in the height of the pandemic, there were two drugs FDA approved for RET fusion positive and RET aberrant thyroid cancers. As I said earlier, RET is activated by two major mechanisms. Non-small cell lung cancer, it's 2%. Papillary thyroid cancer and others, it's 10 to 20%. And RET fusions are seen in less than 1% across multiple tumor types. Interestingly, RET mutations are seen as drivers in medullary thyroid cancer. In a hereditary form of medullary thyroid cancer, such as the MEN syndrome, it's close to almost 100%. In sporadic type of medullary thyroid cancer, it's over 60% harbor the RET mutations. So what are the most common RET fusions? The most common RET fusion partners in lung cancer is the KIF5B RET fusion. The second most common is the CCDC6. In papillary thyroid cancer, the most common RET fusion is the CCD6 followed by NCOA4. And activating point mutations can be seen in the extracellular domain and the tyrosine kinase domain. The most common RET mutations are seen are the M918T. Beyond that, you see the C634F and the entire C series with G, R, S, and Y. So why we needed new drugs for selective RET inhibition? Because we know that drugs such as cabozantinib, vandatinib, lenvatinib, sorafenib, many of the IBs do have RET inhibitory activity. The challenge was the dose reduction rate and the drug discontinuation rates. Because these drugs were not clean and they had like greater potency on multiple off-target kinases like the VEGFR2, they led to a lot of toxicities. Like for instance, if we go up on the dose of vandatinib to inhibit RET, patients, it also tickled the VEGFR2, it also tickled the SRC and other kinases that led to hypertension, hand-foot syndrome, proteinuria, QT prolongation. So this led to drug reduction, dose reduction, and drug discontinuation. So we needed superheroes for RET inhibitors. So you may ask, how do you define superheroes for RET? So if you can see the case of non-small cell lung cancer, if you see the case of osimertinib, crizotinib, ceritinib, the progression-free survival for these agents, for these oncogene aberrant cancers are in the order of 18 months. Whereas with agents like vandatinib, cabozantinib, lenvatinib, the PFS was in the order of 6 months, right? And so we needed new drugs. And another aspect was none of the RET positive cancers, including lung cancer and medullary thyroid cancer, were not as much as sensitive to immunotherapies. We and others showed from our retrospective chart reviews that all patients who harbor RET fusions were not sensitive to immunotherapy. So we needed superheroes to inhibit RET. Let's discuss a case report of a patient. This was a 49-year-old man with advanced medullary thyroid cancer who had progressive disease after six multi-kinase inhibitor treatments over 7 years. The patient received sorafenib, vandatinib, cabozantinib, 2 experimental tyrosine kinase inhibitors. In addition, the patient was on a clinical trial with vandatinib plus everolimus. When the patient presented to my phase 1 clinic, the patient was cachectic, performance status was declining, had 30 bowel movements a day. And a molecular analysis of the circulating cell-free DNA isolated from the blood before enrollment in the vandatinib and everolimus clinical trial identified the founder M918T mutation together with a novel gatekeeper mutation. This is called the RET V804M gatekeeper mutation. And we knew that none of these drugs can target this RET gatekeeper mutation. Although all these drugs could go after this RET M918T de novo mutation, the RET 804M gatekeeper mutation was not targeted with any of these drugs. Enter selpercatinib, or it used to be called LOXO 292. So after we confirmed high kinome selectivity for RET, it was shown in preclinical models that this drug was selective against RET and was better than the multikinase inhibitors, in addition to showing blood-brain barrier penetration. Because the phase 1 study was still ongoing and we needed a higher dose of this drug, we designed an N of 1 study for this patient with an intratumoral dose escalation protocol. Because this was an agent that was only in the preclinical space and it did not enter clinic, we had to design an N of 1 customized study for this patient. Since we did not know the dose, we started at a low dose of 20 based on PK modeling. And we, as you can see, we slowly increased from 20 to 60 to 80 to 120 to 160 twice a day, 160 milligram twice a day. As soon as we hit around 80 milligrams twice a day, we saw resolution of diarrhea and pain within the first week. Calcitonin, the tumor marker, and CA became normalized. Again, as you can see here, there was dramatic drop in the cfDNA, and there was dramatic declines in the tumor markers as well as we went up on the dosing. And you can see a blip here in the tumor marker calcitonin with an 2 asterisks pointed out. That was a major event in Houston called Hurricane Harvey. We had to shut down the clinics, so we couldn't see the patient for 5 days. And the patient had tumor-like, flu-like symptoms and was not doing well at that time. So once the flood receded, we opened clinics. We get the patient back to our clinic. We put the patient right back on treatment, and it started coming down. And that's how sensitive his tumor was for this therapy. This shows dramatic reduction of his liver mass. So this was one of the first patients to be receiving the selective RET inhibitors. You know how I felt? Then I felt like some watcher of the skies when a new planet swims into his skin, or like stout Cortez when with eagle eyes, he stared at the Pacific and all his men, looked at each other with wild surmise, silent upon a peak in Darien. I felt as if I was reading on first looking into Chapman's Homer. So we designed the Libretto 001 clinical trial. And as with any phase 1 study, we started at the low dose and we selected a recommended phase 2 dose of 160 milligram twice a day from this phase 1 study. And as I showed earlier, in the N of 1 study, in the intrapatient dose escalation, we achieved the same 160 milligram twice daily in that single patient. And that is the recommended phase 2 dose. Recently, this was published in the New England Journal of Medicine. And in fact, it was noted as one of the, despite the pandemic, this was noted as one of the most noted publications in 2020 in NEJM, despite all the COVID articles. This showed that the efficacy of selpercatinib was seen regardless of prior chemo, regardless of prior immune checkpoint therapy, regardless of prior multikinase therapy, and the response rate was 68%. Interestingly, these are heavily pretreated patients. And then because of the responses that we saw, we enrolled treatment-naive patients who harbored RET alterations. The response rate in the treatment-naive cohort was 85%. And you can see the dramatic reductions in almost all the patients that were enrolled on this clinical trial. Based on this data set of 144 patients, 105 post-platinum and 39 patients previously untreated, treatment naive, in this rare RET fusion positive non-small cell lung cancer, This combination received the US FDA approval for the treatment of patients with metastatic RET fusion positive non-small cell lung cancer, RET mutant thyroid cancer, and RET fusion positive thyroid cancer. It's important to note that because of the responses seen in the treatment name and every line setting, this was a line agnostic approval. The second agent I'm going to be talking about is the blue 667 or pralsetinib. Similar to selpercatinib, after confirming high kinome selectivity for it and shown that it was more potent and selective than other available multi-kinase inhibitors, the clinical trial was designed. Pralsetinib potently inhibits RET alterations, all resistant mutants, while sparing the VEGFR2. As you can see here, when compared to the other multi-kinases, the blue 667 inhibited RET better. In fact, it went after the gatekeeper mutation as well. We designed this clinical trial called ARROW. This was a first-in-human clinical trial. And based on the PK/PD modeling, we selected an MTD, a recommended phase 2 dose of 400 milligram once daily. It was a once daily dosing. And we showed dose-dependent exposure and RET pathway inhibition of downstream pathways of RET, including RET, downstream MEK inhibition, ERK inhibition, dual phosphatase 6, and SPRODY4 mRNAs. We also saw dose-dependent decline in medullary thyroid cancer tumor markers. Let me share a case of a patient of mine. So this was a 27-year-old young man, right, adolescent, young adult male. Again, he had a massive tumor, a neuroendocrine cancer, thyroid cancer. And this tumor engulfed the carotid artery, internal jugular vessels, and he was brought to us, and they sequenced him and they found a RET alteration. Unfortunately, because of the trachea, the mass that engulfed the trachea, the internal carotid artery, none of the multi-kinases were considered, mainly because he would have a major carotid artery blowout or an internal jugular vein blowout. And radiation was also challenging because it may just lead to tracheoesophageal fistula formation. So we started this patient on a low dose of this agent called blue 667, now known as pralsetinib, at a low dose at 60 milligram and slowly dose escalated. And within 2 to 3 weeks, the patient started feeling better. We saw the tumor shrinkage in this patient. And right now, patient is ongoing at 400 milligram once daily. This young man is taking an oral drug, and it's been almost 3.5 years since he's been on this study. He's gone back to working full time, and his newborn child at the time when he was diagnosed with cancer entered school 3 months ago. And again, these are the stories that keep an investigator, a medical oncologist, motivated, and it motivates us to go to clinic every day and the hospital every day. In fact, in the pandemic, we are just doing telemedicine, local labs, and we are shipping the medications. So he hasn't even set foot into Houston for the last, I think, 1.5 years. And he's taking 4 pills once daily and leading a life close to as normal as possible. This was another story of a young female who benefited from this clinical trial. Again, a mom of 3 kids who harbored a RET fusion positive non-small cell lung cancer. This is another case of a young lady, a 23-year-old woman with papillary thyroid cancer, presented 6 years ago with symptomatic diffuse metastasis requiring supplemental oxygen. Again, since diagnosis, she had required supplemental oxygen, and she was treated with iodine-131 that led to subsequent fibrosis because after the damage from all the tumors and the scarring from the iodine-131 led to fibrosis. Unfortunately, her tumor progressed on that. She progressed on sorafenib and lenvatinib, and she had increasing oxygen needs, developed pleural effusion. Cancer was worsening with declining performance status. She was intubated 3 times over 6 weeks. And it was challenging. And we knew that this tumor harbored the RET alteration. So we, at a window of time when she could be extubated, we started her on this clinical study on Blue 667 at 400 milligram once daily dosing. She had a dramatic symptomatic response. We could wean her from room air to oxygen within 5 months of treatment. And if you see the CT scans here, you see the BMI, and you see the asterisks here. The BMI, she was cachectic and very thin, and with a BMI of 14.8, and it steadily increased in 5 months to 22.3. She remains on treatment now after 3.5 years, started college, completed college, searching for a job. She called us again. We are doing telemedicine and shipping the drugs to her. She's searching for a job and asked a letter of recommendation from me. Again, it was such a pleasure and honor for me to write. Again, we write letters of recommendation, provide references to, so many people. It's so rewarding to write a recommendation letter. I was in tears when I was writing this letter of recommendation for her. Such was her motivation. Again, these are the patients that drive us to find new drugs and, again, motivate us to do what we do. And this ARROW trial moved on. All right. So from the phase 1 study, after selecting the dose of 400 milligrams, and recently the updated data set was published in the Lancet Group journals, and it showed a 73% objective response rate in the treatment-naive RET fusion lung cancer cohort, and 61% in the objective response rate in the post-platinum cohort, and 74% objective response rate in RET mutated MTC, and 60% in those previously treated. Based on this data, this received, again, FDA approval last year. Again, same approval for RET fusion positive non-small cell lung cancer, and this was a line agnostic approval. So you can see the power of precision medicine here. Discovery, preclinical, and rapid clinical validation with registrational studies, all leading to FDA approvals directly from first-in-human clinical trials. Responses were observed regardless of treatment history, regardless of RET fusion partner, regardless of RET mutation or CNS involvement or gatekeeper V8F4M coverage. This is now, we have 2 superheroes for RET. You see the timeline. After it was discovered in non-small cell lung cancer, it's less than 10 years from its discovery, we have not two drugs FDA approved. So what's next? Unfortunately, as cancers follow Darwin's rule of evolution, they acquire resistance. The first patient that I showed you who had a nice dramatic response to selpercatinib, and after 2 years, his tumor started developing resistance mechanisms to selective RET inhibitors. This time was through solvent front alteration called the RET G10S and RET G10C. And these are on-target resistance mechanisms. And as you can see, this solvent front resistant mutants are cross-resistant to both these agents. We tested that in preclinical, and we worked with the structural biologists in the RET models. Selpercatinib and pralsetinib use an unprecedented binding mode to dock into RET kinase. So we needed second generation RET inhibitors that cover these new alterations. So this is on-target resistance. What about off-target resistance? We recently published a patient who developed an acquired NTRK fusion as a resistance mechanism for RET fusion positive lung cancer. And I'm like, this is interesting and intriguing about how cancer overcomes, these resistance. And, it's humbling for us to see, how much we don't know how these cancer develops resistance. Moving to the next topic before I conclude the talk, tissue-agnostic drug development. The sun rises in the new era of tissue-agnostic drug development that is mainly driven by genomics and FDA approvals. FDA approved, this was the first tissue-agnostic approval, right? Pembrolizumab. I think people may not know that. They were thinking larotrectinib is the first one, but this is the first tissue-agnostic approval is pembrolizumab for solid tumors based on MSI high cancers. On May 23rd, 2017, pembrolizumab became the first tissue-agnostic FDA-approved therapy for all cancers that harbor a genomic biomarker. Important to note that this was a tissue-agnostic approval. This was based on a genomic biomarker, and this approval was also based on retrospective and real-world data in addition to prospective data. The second drug was elaretrectinib for NTRK fusion positive adult and pediatric solid tumors. Third drug to receive a tissue-agnostic approval for entrectinib for NTRK fusion adult and pediatric tumors. Recently, last year, pembrolizumab also received histology-agnostic approval for tumors that have a high tumor mutation burden of defined as greater than equal to 10 mutations per megabase. And even more, recently we had the belzutifan for all tumors who harbor a VHL gene alteration. And there was a recent drug that was dostarlimab that was also approved for adult patients with dMMR solid tumors. What about RET, right? What's the next tissue-agnostic drug development, right? So we start, it's beyond a non-small cell and thyroid cancer where it is approved right now. We see RET fusions across across multiple tumor types, although less than 1% in pancreatic cancer and multiple cancers. So we recently showcased that data in selpercatinib across all tumor types like cervical cancer, ovarian cancer, GI cancers, mainly pancreatic cancer. The selpercatinib responses was 47%, and the pralsetinib response rate was 53%. So showing that RET fusions are also a tissue-agnostic target. One thing before we conclude is that, I showed only genomics. The future is going to be everything, right? Microbiomics, proteomics, immunogenomics, transcriptomics, metabolomics. And we need to have a comprehensive molecular portrait of every patient walking in the door. We may not have, I tell my fellows, we may not have a treatment today, but there's always, treatments being developed for every target possible. And the more we test, the more we find. When I started this job, again, one of my fellows, pointed out to me that we didn't have a drug to go after KRAS. Last year, this year we have a drug approved for KRAS G12C. So never say never. And when I started this job in the drug development, when I saw a patient with, say, for instance, BRAF V600 alteration or NTRK fusion, I used to be excited that we have drugs for them. Although we are just still picking needles in haystacks. But whenever I saw a patient who had like 50, 60, 80 mutations, I used to always feel sad, but that there won't be any drug for these patients. Fast forward 8 years, I get excited when I see a patient with 100 different mutations because they are the best patients to receive immunotherapy with a high tumor mutation burden. And so never say never in oncology. There are always new drugs being developed. The death rate from cancer in the US declined by 29%. And the death rate, this was driven mainly by lung cancer. Interestingly, it fell from about 3% a year. And this was attributed to the introduction of novel therapies like immunotherapy, EGFR inhibitors, ALK inhibitors. So right now, we are all engulfed in the COVID pandemic. And now we thought we had a balance in around June, July. And now Delta has put on a difficult strain in our resources. So hopefully we can tilt the balance for cancer care post-COVID era. One of the silver linings of the pandemic, I should say, that when we designed these clinical trials, it was clinical trial centric. But during the COVID pandemic, I think it accelerated the way we design clinical trials to include a more patient-centric interface. And this could be one of the silver linings of the COVID pandemic is designing clinical trials from being clinical trial-centric to patient-centric. And hopefully we take lessons learned from the pandemic to apply to how we design the clinical trials in the future. in summary, I'd like to say that there's going to be an explosion in drug development, and costs will change how early it will be designed, who they will be conducted, and where they will be conducted. Again, we know the cost of conducting clinical trials in the US is very expensive. So more of the clinical trials are being conducted outside the US now. And for precision oncology studies, research, liquid biopsies are coming up big time. And one more important thing is patients will drive clinical research in the future. There's a huge impact of social media, search, and self-driven molecular testing that we are seeing more and more in patients. Again, and thank you to the University of Miami team for the invite. I thank all the patients and their caregivers for participation in all these clinical trials. Thanking all the study investigators and their support and the data monitoring committee, and have all this sponsored support, including from the NIH R01 grant. So please feel free to contact me anytime. This is my email, and I'm available on Twitter as well. Thank you so much. Dr. Wise, I think you're on mute.
Thank you, Dr. Subbiah. Very astute of you, as typically I would expect. That was a beautiful grand rounds. The slides were really informative, and I can tell you, you are really having an impact in the world of cancer. I have had several patients who have gone to MD Anderson with metastatic thyroid cancer and have really reaped the benefits of your work. In addition to phenotyping very carefully the patients, understanding their mutations and eventually their proteomics, lipidomics, etc. How do you go about partnering with the drug developers and getting the next new inhibitor off the shelf and figuring out which one is going to work? Certainly there must be some way in which you communicate with them, that you bring them to clinical trials. Can you enlighten us a little bit about what that process is like and how you interact?
Oh, thank you so much. I think that's a great question. I think in fact one of our chairs has gives us a talk about this and how to interact with sponsors. The thing is, it's a tricky thing, right? So many of the drugs, all the drugs, new discoveries are made in pharma, and we are all in academia. And of course, some of them are in NCI as well. And it's, again, we are still on the learning curve on how we interact, being in academia with all the pharma companies. Having said that, so for instance, for RET inhibitors, again, I started with an investigator-initiated clinical trial. That was one of my first ever studies that I started as faculty when I started in 2012. And back then, after the discovery of RET fusions in non-small cell lung cancer, there were drugs like vandetanib and cabozantinib. So we designed this investigator-initiated clinical trial called vandetanib and everolimus for patients who harbor all RET-positive cancers. So that was available. So patients, not just from MD Anderson, but from everywhere, started coming for this clinical trial. So once we started presenting these clinical trials, we were looking for options for these RET inhibitor clinical trials. So then, there were two companies that approached us on testing the samples and working with them. And we started working much earlier from the preclinical space. In fact, we used to have some of the cancer biologists who used to be working in the RET targeted therapy space. They said, it might be challenging for selective RET inhibitors in the clinic because they didn't expect the preclinical modeling. They didn't expect such great activity with selective RET inhibitors. They were surprised when I used to talk to them. So we interacted with the pharma. We got the drugs to the clinic, and we did rapid phase one studies. And we used to be nimble, agile, and fast in moving these drugs into humans. And it was rapid. I think it was like we worked efficiently with them. And in fact, to open this clinical study, we took just 40 days to open this clinical study in MD Anderson, even the first in human clinical trial, for that particular patient who needed a RET inhibitor at a high dose. We did a compassionate access, and we started the drug within a week, and we moved forward for that. So I think there are different models that we can work. The key is, as clinical investigators, we need to see how we can collaborate with lab people, our own lab people in the in the hospital, and again, the more we collaborate with lab people, the more insights we glean, and networking with pharma companies as well, so that we can get the drugs to our lab colleagues, and they can do the testing, and then we can take it to first in human clinical studies. Thank you so much. I think there's a question from Dr. Rodriguez. Any data for RET inhibitors and immunotherapy concurrently? That's a great question. So we don't have any data yet. So one of the things is what we saw on some of these patients on RET or multi-kinase inhibitor therapies is when they come off, especially if they come off of immunotherapy, and we add a selective RET inhibitor to them, their LFTs go up. We don't know why their LFTs go up, and some of these patients develop some hypersensitivity as well. And this was clearly seen in patients who come off immunotherapy studies. So, and that is one. And two is, as I said earlier, RET positive patients do not respond as well to immunotherapy. So, it's a different topic. So I don't think so. Direct checkpoint combination might be the answer. It's going to be challenging to combine a selective RET with an immunotherapy, although there might be subsets of patients that respond.
Thank you. Any other questions? Please unmute yourself.
So Dr. Kaplan says, how do you approach conversations regarding end-of-life care with so many new developments? Absolutely. I think this is a very important question. So again, we got to be realistic, right? Again, although I showed you cases of the thrill of success in precision oncology, all these patients, respond, and, we are agonized by the agony of failure as well, right? So we have a realistic, honest discussion with patients that, these are, again, the patients are motivated. Patients understand when they come to a phase 1 clinic, they know that they don't have many or any options. So patients do understand, but as oncologists, we need to be realistic in their conversations. In fact, our phase 1 clinic and the palliative care clinic, we call it the supportive care clinic, are next to each other. And we work in close collaboration with them. And, for full disclosure, I may have a major conflict in palliative care because I'm married to a palliative care physician.
Helps a lot.
All right. Well, I thank you again, Dr. Subbiah. We look forward to greeting you in person in Miami when the opportunity arises. I wish everybody a wonderful and safe day. Thanks so much.
Thank you so much. Thank you.